Good afternoon. I am Ritesh and this is my teammates Abhishek and Pranav, and we are working on TNDC particular project, and our project name is brain tumor detection using CNN, SVM and XGBoost.
So basically tumors have plagued mankind from the start of time. And what are tumors? Tumors are basically haphazard growth of cells which result in various types of complications in patient's health. This haphazard growth can be malignant or benign, which means that it can be dangerous to the human body or not dangerous, and depending on which the treatment on patients can be decided by the doctor. So what has happened that diagnosis in previously has been done manually by some diagnostician by doing MRI scans, and during this process there can be many errors caused due to human or manual errors. So as to avoid these, there have been in recent time research in using ML and AI in the field of medicine, so as to reduce these errors and increase in accuracy. And that's why we have today performed tried to read about and perform this project on brain tumor detection. We have used CNN, SVM and XGBoost. We have implemented our project in Spider 5.1.5. In that we have also used Python 3, which is the latest version in the Python. That's why we have used Python 3. Moving forward, this is the GUI that we have made, in which we can use select image. We have the button to select image in order to give image as an input to the code, and then we come to the next part, that is image pre-processing. So in every image algorithm, there's this process called as image pre-processing, in which the image is clarified and basically the MRIs are removed, means extra information that an MRI has is removed, and that's what happens in pre-processing, so as to simplify the process of detection. Then we have CNN prediction, which in the backend has the code to implement the CNN algorithm, and then we have a train model, which is used to train the model of different algorithms. Same goes for SVM and XGBoost model. So next, going forward, I'm going to select an image from the sample dataset that we already have.
So how many samples do you have?
We have around we have used around 9,000 images as a database in the backend, and this is just for the testing purposes, for showing how it works.
From which website you have taken the dataset?
This I have taken it from Kaggle, which is open source database. Which there's also BR35 version, the database name that we have used.
So all the dataset contains the tumor, or
no, not all dataset contains tumor. Some of it contains, some doesn't.
Okay, so to train the model you use both the types of dataset.
Yes, which contain tumor and not.
Okay.
So let's just select an image. Let's see if it's okay. Now we are going to pre-process the image, and this is what has happened. It has converted the image into a grayscale image, so that it can only show the images. Means it can only show that stripped part that has a tumor in it, right? And
okay. Last one is showing the tumors, the white dots.
Yes, so it has stripped away all the unnecessary information and kept the necessary information.
So what kind of measures it has removed from the images?
Sorry?
Under image pre-processing, what is being done?
So it converts it into a grayscale, that is 50.
So only conversion is done?
Yes, and
denoising or
denoising, yes.
What kind of noises are present in that images?
The noises means biologically speaking, the noises which are present are unnecessary cells or unnecessary cells which. And then seen. So this is basically the output that we get after using the system, in which it shows the image testing that has completed, and so it contains a glioma level cell, means glioma level tumor, and the execution time is 1.999 seconds. Moving forward, we can train it using SVM and XGBoost. It does take some time.
So while it is building the model, so it will consider all the data sets which are present in the folder.
No.
How many data sets you are considering for training the model?
For training the model, we have considered all the data sets.
9,000 images.
No, means we have reduced that, but during the initial time we have. So it shows that it has successfully trained the SVM model, and we can see its results in the spider, in which we can see the training score and testing score of SVM model. 86.36 and is the training score, and testing score is 57.196.
Okay.
Now this is for the SVM model.
So it is 86% means what? 86% accuracy or what?
Means
Which percentage it is given?
means
Sensitivity, accuracy, or which feature?
Means it shows us that means what score that tumor they have given it to. Means what is the accuracy? Means it is 86% accurate while training, and while testing it is 57.169.
So how can you increase that accuracy of testing set?
Uh,
So what is the percentage of training and testing data set? That means suppose you have 100 data set or 100 images in the data set.
right.
Out of that, how many are used for the training the model or building the model, and how many images are used for the testing?
Suppose we have 100 for training, we can take it 80.
20 will be used for testing. Okay. What is meant by SVM?
SVM is means basically stand for support vector machine.
Okay. What is the concept behind SVM?
Basically what happens is that SVM works on different types of charting techniques. Means graphic, it is converted into numbers, and it is plotted along the graph, which can be linear and non-linear. So suppose now taking an example where we have red dots and blue dots, and we have to detect whether it is red or blue. So it will chart it along a graph, and it will give a periphery or a periphery or threshold beyond which it will calculate that if it is below this, it will give us blue dots. The output as a blue dot, it is a blue dot, and if it is beyond that, it will give it. Let's say it is a red dot.
So what is the other algorithm you used?
XGBoost.
Can you show the test result for XGBoost?
Okay. We will have to train. So these are the result of XGBoost model that we have trained, in which we have received a training score of 100 and a testing score of 52.27. So which tells us that basically that CNN is superior to both the algorithm among the three algorithms compared to SVM and XGBoost. So these are the results of our project.
So my final question is, so nowadays we actually conclude the brain tumor detection using at least one of the specialists has to conclude that result from the MRI scans.
Right.
Can we use this system as a system to detect the brain tumors or
Yes, mostly we can use it
without any human interface?
Yes, we can use it, but the thing is that it is not implemented still in a large scale where in day-to-day life. It's still a manual-based process. But moving forward, the research has been done extensive. is undergoing extensively, and we can expect that it will be done.
Second question. Which type of tumor it detects? Whether does it detect malign or benign?
Yes, it shows that.
Have you included that in GR? Whether it is non-benignant or non-benignant?
It tells us here whether it is a glioma level or not. Glioma means the tumor that takes place in the glial cells of human body. And,
So what is the future scope of this? What is the novelty of your project?
in this project that we have basically compared different algorithms so as to find out that which algorithms are better, and going forward we can use different types of classifiers in conjunction with CNN so as to see which are better and can be used to increase accuracy.
So try to optimize that.
Yes.